In this video, I'm going to talk about the cellular process of meiosis, which is very similar to mitosis, which you've already learned about. But in meiosis, we're going to take a somatic cell, which is a normal cell found everywhere in your body, and turn it into a gametic cell, or a gamete, which are the cells that are important for sexual reproduction. So what's the difference between a somatic cell and a gametic cell? And the difference really boils down to the ploidy of the cell. So the ploidy of the cell, we'll also use the letter N as a symbol for ploidy, represents how much genetic material is found in the cell. And so there's terms that we use for ploidy. We call a cell that's 1N haploid, we call a cell that's 2N diploid, and we call a cell that's greater than or equal to 3N polyploid. So what does that actually mean? What does it mean, the number of chromosomes or the amount of genetic material? Well, let's look at these drawings of X's. So these X's represent chromosomes that are found in your cell. And in fact, these pairs of X's, one red and one blue, found there, and one red and one blue, are called homologous chromosomes. And they're called homologous chromosomes because they're almost identical. They just have a few differences in those chromosomes. And the reason they're almost identical is because these homologous chromosomes actually come from both of your parents. So in a human being, here's a human being, N is 23. So there are 23 chromosomes that make up an N. So a haploid human cell is 1N. It has 23 chromosomes. A diploid human cell is 2N and has 46 chromosomes. Now, most of the cells in our body are normally diploid, or that 46 chromosomes. And these 46 chromosomes come 23 from our father and 23 from our mother. So we have 23 paternal chromosomes, 23 maternal chromosomes, and that makes a normal cell in our body, which we can also call a somatic cell, makes that cell diploid. It's a diploid cell that has 46 chromosomes. So in contrast, a gamete cell, which is the product of meiosis, we're going to take a somatic cell and make it into a gametic cell, a gamete, only has 23 chromosomes. It's haploid. So a somatic cell is 2N, a gamete is 1N. And I drew this 23 in both red and blue because through the process of meiosis, those 23 chromosomes that make up a gamete are going to be a combination of chromosomes from your father and chromosomes from your mother. And this combination, this variation, is what gives us all of the different genetic combinations that we find in human beings as well as all other living organisms. So ploidy refers to the number of sets of chromosomes. 1 times the number, 23, is haploid. 2 times the number in humans, 46, is diploid. Haploid, diploid. Humans are diploid most of the time. Just our gametic cells, just our sex cells, are haploid. So how do we make these haploid cells? We make them through the process of meiosis. And meiosis is divided into 2 steps, meiosis I and meiosis II, and I'll explain each one on a separate slide, and hopefully the point of each meiosis will become evident as we talk through it. So we start in meiosis I with a cell going through interphase. And since this is a normal cell in your body, a somatic cell in your body, it starts out diploid. N equals 2, it's a 2N cell, it's a diploid cell. As it goes through interphase, this interphase right before meiosis is very similar, almost identical to the interphase right before mitosis. So the cell is going to grow during G1, it's going to replicate its DNA during S phase, it's going to finish up growing during G2, and then it's going to go into meiosis. And specifically, it's going to go into meiosis I. So the first step of meiosis I is called prophase I. The same terminology that we used in mitosis, and the same general ideas happening in prophase I. Our nuclear envelope is disintegrating, and our genetic material, our chromatin in interphase, is condensing into chromosomes. And you'll see here that I've used the traditional X's for chromosomes, but I've added another set, and I've just used A for clarity and graphic. So there's no actual chromosome in a microscope that looks like an A, but we'll just use the A because that's another letter that we can look at so we can distinguish what's going on. So you'll notice we have both an X and an A chromosome from the father, and an X and an A from the mother. So this gives us a diploid cell. We have two sets of chromosomes, two sets of AX, a blue AX and a red AX. So in prophase 1, after they've condensed, we'll move on to metaphase 1. And metaphase, just like in mitosis, is going to result in chromosomes aligning at the metaphase plate in the middle of the cell. And they're going to align by the spindle fibers attaching to those chromosomes and pushing them and pulling them until they're lined up perfectly in the middle. Now, the difference between metaphase 1 of meiosis and metaphase and mitosis is that in meiosis 1, we're not going to line up single chromosomes in the middle. Instead, we're going to line up homologous chromosomes. So these homologous chromosomes are the chromosomes that are almost identical to one another. And you can see our homologous chromosomes include our A and A, our maternal and our paternal A will line up, and our paternal and our maternal X will line up next to each other. So these are known as homologous chromosomes, or also tetrads. So in metaphase 1 of meiosis 1, these tetrads will align at the metaphase plate. Following metaphase, we'll go to anaphase 1. And in anaphase 1, we will separate the homologous chromosomes. So we will now pull a full chromosome to each side, a full A to that side, a full A to that side, a full X to that side, a full X to that side. So we have not separated chromatids at all. We are only separating homologous chromosomes in meiosis 1. Following anaphase 1, we go to telophase 1, where the nuclear envelopes start to reform. The nucleic acid, the chromosomes in telophase 1, can decondense in some cells. However, in other cells that proceed to meiosis 2 very quickly, these don't actually condense here. So for our purposes, we can say that these are not condensing because we're going right into the next step after this. So the nuclear envelope starts to reform in telophase, and then we get to cytokinesis. So we've now divided into two cells. Now, following meiosis 1, our two cells that we end up with are now haploid because they have an N of 1. So now we only have one copy of A in this cell and one copy of X in this cell. Same here, one copy of A, one copy of X. So these two cells are haploid. So following meiosis 1, we have two haploid cells. However, we haven't completed the process of meiosis to make gametes. And the reason we need to do more steps is that we still have sister chromatids left in these cells. So to separate the sister chromatids, we'll move on to meiosis 2. And if you understand what's going on in meiosis 1, I think meiosis 2 is actually even simpler to understand. Because meiosis 2 is almost identical to what we looked at in mitosis. Because now in meiosis 2, we start with prophase 2, where our nuclear envelopes are going to start disintegrating. We move to metaphase 2, where our chromosomes, single chromosomes, are going to align along the metaphase plate. After metaphase 2, we move to anaphase 2, where we will actually split the sister chromatids apart. So we are now going to pull one half of the X this way and one half of the X that way, one half of the A this way and one half of the A that way in anaphase 2. So we've now split the sister chromatids in anaphase 2. We move to telophase 2, where we start reforming our nuclear envelopes and decondensing our chromatids. And we end with cytokinesis, which will make 4 gametes by the time we're done. Because we started meiosis 2 with 2 cells, each cell in meiosis 2 divides in half. We now have 2 times 2, 1, 2, 3, 4 cells, 4 gametes that we end up with at the end of meiosis 2. Now, compared to the beginning of meiosis, these cells now have one copy of one half of the genetic material that we started with, because we've divided that genetic material in half in meiosis 1, and then split that material in another half, made one copy go to each cell in meiosis 2, and that gives us 4 gametes. Now, gametes is the generic term, gametic cells is the generic term we use for sex cells. And in humans, and in fact many organisms, the male gamete is known as a spermatid, or a sperm cell, and the female gamete is known as an ovum, or an egg cell. Now, because of the process of meiosis, female gametes end up with one ovum and 3 polar bodies, which are smaller cells that are not used for sexual reproduction. And we'll talk about in a second for fertilization exactly what happens between the spermatid and the ovum. But the polar bodies are there because the ovum needs to have as large of a cytoplasm as it possibly can. So the polar bodies are split-offs of chromatin and genetic material that is not going to be used for any further parts of the reproductive process. So once all of these gametes are made, once we have sperm cells and we have egg cells, we go to the process of fertilization. So here's where we take 2 haploid cells, we take a sperm cell and an egg cell, and we combine them to give us a zygote, which is now a diploid cell. So this is the life cycle of organisms that reproduce sexually. They go through meiosis to create gametes, so they go from diploid to haploid, and this is true for humans. They go from diploid to haploid to make sex cells, to make gametes. Those gametes then recombine to make another diploid. In this case, we're going to call this diploid cell a zygote, and this zygote will then proceed to mitosis, and it will go through many, many rounds of cellular division, of mitosis, until it becomes a fully sized human being, at which point it will begin making more gametes by meiosis and repeat the process over and over and over again. Now, I mentioned about those polar bodies, and I mentioned that the ovum needs to stay as large as it possibly can. And that's because the zygote, the cell that makes up the zygote, is primarily composed of the material that's within the ovum. So all of the things that a cell needs to survive, the mitochondria, the Golgi, the endoplasmic reticulum, the ribosomes, in fact even some mRNAs, remain in the ovum. And the male gamete, the sperm cell, just adds a little bit of nucleic material to make this cell diploid. This cell is going to continue on through life utilizing those mitochondria, the endoplasmic reticulum, the Golgi, the ribosomes that came from the maternal lineage because of that. So just to review briefly what we're seeing here, mitosis, this cell is proceeding to mitosis, which is 2N to 2N. So once this zygote goes through meiosis, it's just going to keep replicating and making the same cell over and over and over again. And in fact, mitosis works from any N to any N. So if you have a 1N cell and it goes through mitosis, it's going to end up with 2 1N cells. If you have a 3N cell, a 4N cell, you're going to go through mitosis and end up with 2 3N cells or 2 4N cells. So mitosis is always concerned with ending up with the same number of chromosomes that you started with. Meiosis, on the other hand, is going to take us from 2N to 1N, or it's going to take us from diploid to haploid cells. The diploid cells are also known as somatic, and the haploid cells are known as gametes. And we'll follow up meiosis at some point with fertilization, which is going to take us from 1N back to 2N, or haploid back to diploid. And so this cycle between meiosis and fertilization allows us to go from haploid back to diploid, diploid back to haploid. We'll go through meiosis, go through fertilization, go through meiosis, go through fertilization. And this will become important when we talk about genetic differences between organisms. This is the crux of where genetic differences come from. So to just give you a final overview of what we've talked about for meiosis, an important term we learned about meiosis is ploidy. Ploidy relates to the number of chromosomes that are found in the cell. So a 1N cell has 1 copy of a chromosome. That makes it haploid. And we called these haploid cells gametes. A cell that's 2N has sets of homologous chromosomes. So it has 2 copies of chromosomes. And we call those cells diploid. And these diploid cells in our bodies are normal, and we call them somatic cells. The last thing about ploidy that I alluded to is that if you have more than 2, greater than or equal to 3 sets of chromosomes, it's referred to as polyploidy. You can use terminology such as tetraploid, triploid to describe that, but polyploid is a generic term meaning more than 2 sets. It's quite normal in some organisms. In fact, many plants are polyploid by their nature. It's not abnormal. It's how they go through life. But in human beings, polyploid cells are a hallmark of cancer cells. We've talked in class about the HeLa cells that are used for research. HeLa cells actually have more than twice the normal number of chromosomes that a cell should have. And scientists believe that part of the reason that those cells are so cancerous is because of the multiple ploidy that those cells have. We're going to call meiosis reductive division. And the reason we're going to call it reductive division, and the thing I want you to remember about meiosis, is that meiosis takes a cell that's 2N and makes it 1N. So we're going to go from diploid to haploid whenever we go through meiosis. Now specifically when we go through meiosis I, we're going to align and separate our homologous chromosomes. So meiosis I separates homologous chromosomes. Then we're going to go to meiosis II, which looks like mitosis, where we're going to align and separate our sister chromatids. So meiosis I separates chromosomes, meiosis II separates chromatids. At the end of meiosis II, we're going to have gametes, we're going to have haploid cells, and these haploid cells will recombine in fertilization. So we're going to recombine our gametes to create zygote. The zygote is the new diploid cell that we get by combining 2 haploid cells. So if you can remember that meiosis allows us to go from a diploid 2N cell to a 1N cell, and the details that meiosis I separates homologous chromosomes and meiosis II separates sister chromatids, you should be all set for understanding the important process of meiosis.